Hello my dear lovely students. So once again welcome back on this amazing platform of Physics Wallah, where I, Ayushi Agarwal, your botany teacher, is going to teach you the next important chapter of class 11th, that is respiration in higher plants. This chapter is very easy, simply conceptual chapter. If you once understand the reactions going on during the breakdown of glucose, then this chapter is extremely easy as well and very scoring as well. Usually students get scared with this chapter. Yes or no? We do get scared when the questions of respiration are asked, reaction based questions, that in which step what is going to happen, what is the substrate that is going to be produced, and so many questions. But students, if you listen to this lecture carefully as well as clearly, then surely you will be able to answer all those questions. See, once you understand the concept, then learning becomes easy. Learning you have to do. But if you first understand and then you try to learn, then surely the things are going to become better and easy. So I hope that you're going to enjoy the whole lecture, and please do not miss any of the section, because this chapter specifically, all the subheadings in this chapter are correlated to one another. If you miss one heading, then your connection with the next heading is also going to be missed. So if you are watching this session from the beginning, try to continue till the end. You may take certain breaks, but do not leave them in between or do not jump the video. So simply, you want to fast forward it, do it, but if you start the lecture, then please go till end also. Stay connected with all the subtopics, and then you see how interesting this chapter is going to become. So our chapter is respiration in higher plants. Let's start with this chapter. Okay, so first, as usual, comes your introduction part. In introduction, you should know what do you mean by respiration. So respiration is simply, it's a chemical process. So respiration can be defined as a chemical process by which the food which you intake is broken down into its simplest form in order to release energy. And this breakdown of your food is a multi-step process. Each step is controlled by some of the different enzymes, right? It is a chemical process as well as it is irreversible, because the food once which is broken down cannot be rejoined to form your original food once again. So hence, respiration is a chemical process. It is an irreversible process where the food, for example, the food is sugar. Right, so the food in the form of sugar is having so many bonds, complex carbon-carbon bond, carbon-hydrogen bond, carbon-oxygen bond. So during respiration, all these bonds are broken down to produce simplest products. While doing this, while the bonds are being broken down, the energy stored in this bond are released in the form of ATP. That ATP we are using in our metabolic activities. Plus, it is not a single step process, rather it is a multi-step process, and each step is under the control of some or the other enzyme, right? So let's check out the definition, what is written on the screen. Respiration is a chemical process, irreversible process by which food is broken down to produce energy and in a multi-step process, correct? Now, sometimes we do get confused between respiration and breathing. Sometimes we consider that these two terminologies, breathing and respiration, are synonym to each other. But my dear students, let me tell you for your good information that these two terminologies are not synonyms. Breathing is simply exchange of gases, the inhalation and exhalation. Now, what happens when you inhale the gas? Oxygen is coming in. That oxygen is used for respiration, for the breakdown of food. And during the breakdown of food, whatever end products like CO2 and water are generated, then that is exhaled out. So simply intake and outtake of gases is breathing. Now whatever the remaining process with the food is taking place, that is called as breathing. So that is called as respiration. So breathing and respiration are two completely different processes. You can say yes, that if breathing is taking place, then only gases are being exchanged and that gases are being utilized for respiration, and whatever gases are generated during respiration is released out with the help of breathing. So they are like functionally they are correlated, but you cannot interchange these terminologies. I cannot say that energy is generated in my body due to the process of breathing. No. Right? Or I cannot say that respiration is inhalation and exhalation of gases. No. These two are not synonyms, but yes, these two different processes are helping each other to function. Right? Plus, breathing is a physical process. It is reversible, but respiration is a chemical process and it is irreversible. So these two terminologies are different. Clear? Very good. Now, some more points related to breathing, with respect to respiration, is that it is an amphibolic pathway. Respiration is neither catabolic nor anabolic. It is rather amphibolic pathway. Means it is both catabolic and anabolic. Now I'll explain what is amphibolism in detail when I'll proceed in the chapter. So for time being, you should know that respiration is an amphibolic process. Amphi means both. What both? Both catabolic and anabolic. Correct? How? That I will tell you later in the chapter. It is an exothermic process. That means during this process, energy is being released out. You know, the food, the complex food stores energy. And when you break down that food, then lot amount of ATP is generated, which you use for your metabolic activities. Hence, respiration is amphibolic, exothermic, chemical, irreversible process. I hope all the terms related to respiration so far is clear. Okay. Now, I am saying that during respiration, food is broken down. The food which is broken down during respiration is given a specific term. And that term is respiratory substrate. So, whatever food is being broken down during respiration, maybe fats, maybe carbohydrate, sometimes even proteins and organic acid are also broken down in extreme conditions. So in that case, those food particles which are broken down to produce energy are known as respiratory substrate. Clear? Okay, now respiration, depending upon the types of respiratory substrate, that which type of food is being broken down, respiration can be of two types: floating respiration and protoplasmic respiration. If I talk about floating respiration, then what do you mean by floating respiration? Floating respiration is the most common type of respiration which usually takes place in our body. That is when carbohydrates or fats are used as respiratory substrate. Correct? So what is floating? Floating respiration means when carbs or fats are used during respiration as respiratory substrate. Clear? Second is protoplasmic. Protoplasmic simply means when the respiratory substrate is either protein or organic acid, maybe like malic acid, oxalic acid, etc. Correct? So, what is protoplasmic respiration? When respiratory substrate is either proteins or organic acid. Now, Floating respiration is the most common type which in generally takes place. Right now, whatever energy is being generated in my body is due to the breakdown of carbohydrates, maybe fats. So in usual condition, the type of respiration taking place is floating. But suppose I am starving. I am not getting food since 10 days, not even a single bite of food. I am starving. So what will happen? For 7 days, suppose if I am not getting the food, then all the carbohydrates and fats which are right now stored in my body will get dissolved. But still the body wants to survive. In that extreme case, when all the carbohydrates and all the stored fats are over in my body, then body in order to survive will start utilizing proteins. It will start taking out the proteins from the cell membrane, from the protoplasm of the cell, thus killing the cell. Now imagine if you break down the cell membrane, if you remove the protein from the cell membrane, then what will happen? Membrane will break down, thus resulting in the lysis of the cell. So you can say that this protoplasmic respiration, if it is taking in your body, then surely it is an alarming situation. You are surely in great need of food. Correct? So right now in my condition, I'm happy, I'm eating my food daily, I'm full of carbohydrates and fats, so my body is easily performing floating respiration. But suppose no fats, no carbohydrates, not eating food since 10 days, starving, then in that case my body will start utilizing proteins in their protoplasm and in the cell membrane. That means my body is degrading and it is an alarming situation. Correct? So that means what will be the second point of difference? Floating respiration, it is occurring usually when the body is rich in carbs and fats. Occurs majorly when the body is rich in fats and carbs. However, under extreme condition, when you are not eating any food, under extreme condition like starvation, condition like starvation, when all carbs and fats are over from the body, then body in order to survive start utilizing proteins from the protoplasm, cell membrane, etc. So under extreme condition, protoplasmic respiration operates. Correct? Okay. Now moving ahead. Again, there can be two types of respiration on the basis of requirement of oxygen. The two types of respiration on the basis of oxygen is aerobic and anaerobic. Very easy. You all know what is aerobic and anaerobic. But still, let's have a quick look on these two types of respiration. So what is aerobic respiration? The breakdown of sugar, the breakdown of respiratory substrate, if it takes place in the presence of oxygen, right? Anaerobic means when the breakdown of respiratory substrate takes place in the absence of oxygen, then it is known as anaerobic respiration. First point of difference. Second, my dear students, if aerobic respiration is getting operated, then the respiratory substrate is going to get completely broken down. If I talk about carbohydrate, which is the general most respiratory substrate, so if carbohydrate like glucose is broken down in the presence of oxygen, then in that case it will be completely oxidized into the simplest form, which is CO2 and water. So under aerobic condition, complete breakdown of sugar, the most common respiratory substrate, takes place. And what are the end products in that case? the end products will be CO2 and water. Right? Now, these two end products, they are non-accumulable and non-toxic. These two end products will be soon released out during exhalation. As soon as they are produced in the body, you're going to exhale like this, and the entire CO2 water is removed. So you can say that end products are non-toxic as well as non-accumulable. They do not accumulate. They are soon released out as they are produced. Accumulate. Right? Now, of course, when complete breakdown is taking place, all the bonds are being broken down of the sugar, then surely the amount of energy produced will also be high. So in case of aerobic respiration, the amount of energy generated per glucose may be like 36 or 38 ATP. Correct? Now, coming to anaerobic respiration. First of all, second point, in case of anaerobic respiration, there is partial breakdown of respiratory substrate. Right? As a result, instead of end products being CO2 and water, they can be either alcohol or lactic acid. So end products are not usually CO2 and water, rather it can be like alcohol or lactic acid. Right? Now these end products, they are accumulable. Like when you do excessive exercise, then your muscle cells, they start performing anaerobic respiration. They start producing lactic acid. And you know, when lactic acid deposits in your muscle cells, then they cause muscle fatigue and pain for certain days. Why? Because they accumulate in the body. As soon as they are produced, they are not discharged. And because of their accumulation, pain is being generated. So you can say they can be they are accumulable as well as they can be toxic. If I talk about alcohol, usually yeast cells, they perform alcoholic fermentation, they perform anaerobic respiration. And if this alcohol produced in yeast cells starts getting accumulated, and if the percentage in their body reaches beyond 13%, then it can also lead to the death of yeast cells. So we can also say that these end products can be toxic as well as accumulable. So what you should write, they can be toxic as well as accumulable. Now, since there is partial breakdown, so obviously less number of bonds are broken down. As a result, total amount of energy produced also becomes less, which is very less, equivalent to only 2 ATP. Right? So these are some basic points of difference between aerobic and anaerobic respiration. I hope these things are also clear in everybody's mind. Okay. Now, you have seen that there are two types of respiration on the basis of oxygen requirement, aerobic and anaerobic. Now, this chapter is all about understanding in detail that what happens during aerobic respiration and what happens during anaerobic. So, we are going to take the very common respiratory substrate glucose, and we will break down this glucose in both the ways, anaerobically and aerobically, to understand that how ATP is generated and what are those steps. Correct? So now we are going to study both the types of respiration by using the substrate glucose, and let's start. Okay. So my dear students, the initial step of breakdown of glucose is common in both aerobic as well as anaerobic respiration. Those initial steps for the breakdown of glucose that is common in both aerobic and anaerobic together is termed as glycolysis. So what is glycolysis? Glycolysis is the first pathway of the breakdown of glucose, which is common in both aerobic and anaerobic respiration. First point. Second point, glycolysis means what? Basically, this glycolysis word is derived from a Greek terminology that means sugar splitting. Glyco means sugar and lysis means splitting. Right? So during glycolysis, we are going to simply break down the sugar. Which sugar? Glucose. The simplest form of sugar, glucose, a 6-carbon compound, during glycolysis is broken down into 2 molecules of 3-carbon pyruvic acid. Right? So you are having the most common respiratory substrate in your hand, glucose, to which you have to break down into 2 molecules of pyruvic acid, and this breakdown is not a one-step process. Rather, So as I'm telling you, this breakdown is not a one-step process. Rather, to perform this, 10 steps are required. And each step is controlled by some or the other enzymes, and these 10 steps altogether is termed as glycolysis. It is common in both aerobic and anaerobic. That means whether it is aerobic or anaerobic, pyruvic acid will be produced in both the cases. Right? This process occurs in the cytoplasm of the cell. Where does this process takes place? In the cytoplasm, in the absence of oxygen. So we can say that glycolysis is actually an anaerobic process. That is why it is common in both aerobic and anaerobic. Right? Okay, glycolysis, my dear student, the 10 steps were discovered by 3 scientists, Embeden, Meyerhoff and Parnas. That is why glycolysis is also known as EMP pathway. E stands for Embeden, M stands for Meyerhoff and P stands for Parnas. So the name of the 3 scientists who discovered, their initials were joined and the another name for glycolysis was proposed. Right? Okay. Now the 10 steps. Main focus is to understand that what are those 10 steps and how glucose is converted into pyruvic acid. I hope all the points which I have written so far, 1, 2, 3, 4, 5 is clear. 6th point I will tell you while writing the entire pathway of glycolysis. Shall we start glycolysis? Okay, so buck up your seats everybody, and let's start with glycolysis, events that takes place during glycolysis. My dear students, the 10 steps which I have mentioned is usually divided into 2 phases called activation phase and payoff phase. Now what is this activation phase and what is this payoff phase? I'll talk about while teaching you the events. Okay, so the first step in glycolysis is you are having glucose. But if I talk about plant cells, see, food is produced, right? Now, when it comes to respiration, when photosynthesis takes place, okay, so food is produced in the form of glucose, stored in the form of starch and transported in the form of sucrose. So usually cells, they receive sugar in the form of sucrose. Now the sucrose can be broken down, it's a disaccharide which is then broken down into its 2 substrate, that is glucose and fructose. Right? So glucose and fructose both are ready to undergo glycolysis. So if you are provided with sucrose, so understand that sucrose is a disaccharide which is made up of 2 sugar components, 2 monosaccharides, glucose and fructose. be it glucose, be it fructose, both are ready to undergo glycolysis event very fast. There is no issue that only glucose will enter or fructose. No, both are absolutely ready to enter into glycolysis. So here I am taking glucose as my respiratory substrate. Even if the question is asked for sucrose, then you should know that sucrose will also undergo glycolysis, but first it will break down into its monosaccharide, glucose and fructose, and these two separately will then join the glycolysis. Right? So first I'm going to start my pathway by using a 6-carbon compound, glucose. Right? Now I am having glucose as my respiratory substrate. The first step will be phosphorylation of glucose. See, glucose first need to be made ready to undergo breakdown. To undergo oxidation, this glucose has to be made ready. And for making glucose ready, you have to spend, the cytoplasm of the cell has to spend energy. So initial steps of glycolysis, they require ATP, they consume ATP in order to activate glucose. Once glucose gets activated, then it starts producing ATP. That is why this glycolysis is divided into 2 phases. Activation phase. This phase counts for those steps of glycolysis where energy is given to the substrate so that it can become ready for glucose, for ATP production. And once your substrate gets activated, once it is ready to release energy, then the remaining steps are known as payoff phase, where energy is produced. Correct? Chalo. So let's start with first initial steps are activation phase, and later on steps together are payoff phase. So first step is activation of glucose by providing it with ATP. Okay, so one molecule of ATP will be broken down. It will be to release ADP, an inorganic phosphate. This process is taking place in the presence of the enzyme hexokinase. So hexokinase enzyme will phosphorylate glucose. How? By adding this inorganic phosphate to the 6th carbon of glucose. Right? You know glucose is a 6 carbon compound. So to the 6th carbon, this phosphate group will be attached. And what will be the intermediate in that case? The intermediate will then be glucose 6-phosphate. So first step is phosphorylation, where energy is consumed in activating the glucose and you produce glucose 6-phosphate. Now this glucose 6-phosphate in the next step will undergo isomerization. The functional isomer of glucose is fructose. So glucose 6-phosphate with the help of the enzyme isomerase will convert into its isomeric form, that is fructose 6-phosphate. Right? And which enzyme is required? Isomerase enzyme is required. This is also a 6-carbon compound. Obtain fructose 6-phosphate. Now in the next step, fructose 6-phosphate further wants to get activated. Still activation phase is getting continued. So again, in order to activate this intermediate, one more ATP will be broken down. One more ATP will be broken down, and this time the free phosphate that is released will now be attached to the first carbon of fructose 6-phosphate. And what will be produced? The substrate, the intermediate produced will be fructose 1,6 biphosphate. Right? Already on 6th carbon phosphate is attached. Now on 1st carbon also phosphate will get attached. So on fructose, a 6 carbon compound, 2 phosphates are attached, 1st and 6th. So position I have mentioned, and accordingly the name of the substrate is here. And what will be the enzyme required in that case? In the 3rd step, enzyme required is phosphofructokinase. Correct? So with the help of the enzyme PFK, activation of fructose 6 phosphate takes place to produce a 6 carbon compound, fructose 1,6 biphosphate. Clear? Now this 6 carbon compound, my dear students, it will now undergo splitting. So next step in glycolysis is splitting of this 6 carbon compound to produce 2 compounds, each having 3 carbon. So in the presence of the enzyme aldolase, which enzyme? Aldolase, this 6 carbon compound will break down into 3 carbon compound each, called phosphoglyceraldehyde and dihydroxyacetone phosphate. Correct? Yes or no? Till here is your activation phase, because till here you have spended energy, you have given in something to the glucose, you have not received anything. Till now you have spent 2 ATP molecules in the first step, and second and the third step. So you are providing something to the substrate, so that substrate can get activated. So therefore the initial steps are known as activation phase. Activation phase is done. Now let's proceed with the payoff phase. Now before payoff phase starts, this DHAP, it converts. Now DHAP and PGAL, they both are isomers of each other. So here also isomerization will take place, and this DHAP will convert itself into PGAL, again with the help of the enzyme isomerase. So already PGAL was one molecule, DHAP also converts into PGAL. Hence how many molecules of PGAL now becomes? 2 molecules of PGAL are produced. So till here, as I told, is your activation phase. Now whatever happens with the 2 molecules of PGAL will come under payoff phase. I hope everything, all the enzymes, all the steps are clear. Now we are going to start with the payoff phase. So now what you are having? You are having 2 molecules of phosphoglyceraldehyde, right? Now these 2 molecules of phosphoglyceraldehyde, my dear students, will undergo oxidation. Oxidation in the absence of oxygen. Then how? By removing hydrogen and electron. So from PGAL, oxidizing agent NAD positive will come. Already cytoplasm is rich in all these oxidizing agents. So one of the oxidizing agent, famous oxidizing agent, NAD positive will come, will oxidize PGAL, will remove its proton and electron and will oxidize it. Along with oxidation of PGAL, phosphoric acid will donate its phosphorus as well. Right? So 2 things are going to take place with this 2 molecule of PGAL. 2 steps. Now you see what are those 2 steps. In the first step, it will be oxidized. Oxidation with the help of oxidizing agent NAD positive. So NAD positive, one NAD positive will remove 2 proton and 2 electron from PGAL, and itself it will get reduced to NADH2. So oxidation is done, correct? 2 molecules of PgAl are there. So 2 molecules of NAD. If 2 molecules of NAD, then 4 proton and 4 electron. Right? So let me just write 4 proton and 4 electron. Reduced NADH2 is produced 2 molecules. Along with oxidation, this PgAl will also add phosphate group, but no ATP will be consumed because now we have started payoff phase. So no ATP. So from where phosphate is going to come? So phosphoric acid in the cytoplasm will come to donate its phosphorus. So after phosphorylation and oxidation, 2 molecules of phosphoglyceraldehyde will get converted into 2 molecules of 1,3-biphosphoglyceric acid. So what will be produced after this? 2 molecules of 3 carbon compound 1,3-biphospoglyceric acid. Right? Now, these 2 molecules of 1,3-biphosphoglyceric acid will now pay off, will now release energy. How? Now, the first phosphate, can you see? On the first carbon of this biphosphoglyceric acid, phosphate is attached. This phosphate will now be removed and it will be given to ADP. So 2 molecules of ADP will come. Right? 2 molecules are there. So 2 molecules of ADP will come. They will remove the first carbon phosphate. What they will produce? They are going to produce energy. So this is the first level, this is the first reaction, this is the first step in glycolysis where you are getting direct energy. And therefore this step is known as substrate level phosphorylation, because with the help of substrate you are phosphorylating ADP in order to produce ATP. Correct? And in return, these 2 molecules will convert themselves into 3-phosphoglyceric acid. Right? Now, this step is known as substrate level phosphorylation. Why? Because with the help of substrate, we are adding phosphate to ADP in order to produce ATP. Right? Okay, first phosphate has been removed, but still this compound is having phosphate on its third carbon. So the substrate is 3-phosphoglyceric acid. Now, after obtaining 2 molecules of 3 carbon compound 3-phosphoglyceric acid, next step will be rearrangement. Rearrangement what? 3-phosphoglyceric acid will convert into 2-phosphoglyceric acid. Okay, one enzyme I have forgot to tell you, the enzyme which is required in this oxidation step. This enzyme name is important and you should know. The enzyme name is glyceraldehyde dehydrogenase. Dehydrogenase. Wherever enzyme name is required, I'll keep on telling you. Right? So now you have obtained 2 molecules of phosphoglyceric acid. Now these 2 molecules of phosphoglyceric acid will undergo dehydration in the presence of the enzyme enolase. So enolase will dehydrate, will remove water from PGA and will produce 2 molecules of phosphoenol pyruvate. So now what are we going to receive? 2 molecules of phosphoenol pyruvate. It is also a 3 carbon compound. Correct? Finally, this is the 9th step. Now last 10th step. Phosphoenol pyruvate will again undergo substrate level phosphorylation, will remove its phosphate, will give it to ADP to produce ATP, and itself will convert into the final product of glycolysis, that is 2 molecules of pyruvic acid. Here also 2 ATP will be generated, and this step is taking place in the presence of the enzyme pyruvate kinase. So last step also ATP will be produced. So this step is also substrate level phosphorylation to produce 2 molecules of 3 carbon compound pyruvic acid, and this is also taking place in the presence of the enzyme pyruvate kinase. Clear? I hope all the 10 steps are clear to each and everybody. So can you see that how we have obtained from 1 molecule of glucose 2 molecules of pyruvic acid with the help of 10 steps. Different steps are controlled by different enzymes. Initially you have to provide energy as it was an activation phase, and then later you obtained energy along with NADH2. So all the steps which are written on this page, they come under payoff phase, and all the steps which are written in this page, they all are of activation phase. Right? I hope all the steps you have understood so far, that what is all about glycolysis. Now, end result of glycolysis. What actually you obtained, what you gave and what you obtained. Okay, so we provided 1 molecule of glucose in the cytoplasm of the cell, and what we did, this 6 carbon compound was finally broken down into 2 molecules of 3 carbon compound pyruvic acid. Meanwhile, converting 6 carbon into 3 carbon compound, what all you obtained and what all you gave. So what we gave only 2 ATPs. 1 ATP you gave at the first step, and second molecule of ATP you gave at the third step. Right? Apart from this, what you obtained? So you gained 2 molecules of pyruvic acid. Apart from this, you obtained 4 ATP. How? There were 2 substrate level phosphorylation reactions. See, 2 ATP over here and 2 ATP over here. So total you gained 4 ATPs, then 2 molecules of NADH2 and 2 molecules of water. So these are the things that you obtained. 4 ATP, 2 NADH2 and 2 molecules of water along with 2 molecules of pyruvic acid. Right? Now, if I have to calculate the net gain ATP in case of glycolysis, the net gain, total produced ATP is 4. If the question comes how many ATPs are produced in glycolysis, then your answer will be 4. But if the question comes what is the net gain, then net gain of ATP will be 4 minus 2 ATP. So net gain of ATP during glycolysis is 2, because 2 we also initially give to activate glucose. You obtain 4, so remaining is 2. I hope the product of glycolysis as well as the reactions in glycolysis is clear along with the result. Now my dear students, I told you that glycolysis is a common respiratory pathway in both aerobic and anaerobic respiration. It is done. Pyruvic acid is obtained. Now, whether oxygen is there in the cell or not, that will decide the fate of pyruvic acid. If oxygen is there in the cell, then this pyruvic acid will enter into the mitochondria for further breakdown. That will be called as aerobic respiration. However, if your cell does not have oxygen, if conditions are still anaerobic, then your pyruvic acid will remain in the cytoplasm and will undergo anaerobic respiration. That means the fate of pyruvic acid produced after glycolysis depends upon the availability of oxygen. Hence, our next topic will be fate of pyruvic acid. We have obtained pyruvic acid from glucose. Now let's see how this pyruvic acid will undergo further breakdown under the heading fate of pyruvic acid. So first I'll be teaching you the condition where oxygen is absent. That means first we are going to study anaerobic respiration, that fate of pyruvic acid if the conditions are anaerobic, and then I will teach you fate of pyruvic acid if in the cell oxygen is provided. Right? So let's start with anaerobic respiration. First is fate of pyruvic acid in the absence of oxygen. Right? In the absence of oxygen, that means anaerobic respiration, also called as fermentation. So first we are going to study fermentation. Fermentation can be done by two ways: lactic acid fermentation and alcoholic fermentation, depending upon that what are the end products of pyruvic acid. Correct? So first I'll be teaching you the first type of fermentation, that is alcoholic fermentation. Usually alcoholic fermentation is done by organisms like yeast. Right? It is usually a two-step process. Two-step process where pyruvic acid, okay, fermentation, my dear, takes place in the cytoplasm only. So pyruvic acid is also there in the cytoplasm. This pyruvic acid need not to go anywhere. It will remain in the cytoplasm and will undergo fermentation. First is alcoholic fermentation. It is a two-step process where pyruvic acid will be reduced. Fermentation chemically is reduction in nature. It is chemically reduction reaction. So here pyruvic acid in two-step process will be reduced to alcohol and CO2. Right? For reducing, the reducing agent, should be there. So reducing agent will be NADH2, the one that you produced during glycolysis. See, during glycolysis we produced 2 NADH2, right? So if this pyruvic acid has to undergo fermentation, for reducing it is going to use the same NADH2. Correct? Okay. So reducing agent is NADH2, in bracket I will write produced during glycolysis. Right? Now let's see the reaction, the two-step reaction. First reaction is decarboxylation. So first reaction of alcoholic fermentation is decarboxylation. See, this is your pyruvic acid. Right? What will happen? This pyruvic acid will first undergo decarboxylation, matlab removal of CO2 in the presence of the enzyme pyruvate decarboxylase. So in the presence of the enzyme pyruvate decarboxylase, this pyruvic acid, now this CO2 will be removed. Can you see? The CO2 will be removed. What will be remaining? CH3CHO. See this? Acetaldehyde will be produced from pyruvic acid. So in the first step you obtain acetaldehyde. Now what will be the second step? Whatever acetaldehyde is produced, now this acetaldehyde, my dear students, will undergo reduction, and the reducing agent will be NADH2. Two molecules are there. Oh, what I wrote. Right? Now, two molecules of CH3CHO is there. So here also you will write second step, two molecules of acetaldehyde, two molecules of CO2. Right? Now what will happen? One second. It's so difficult to use smart boards. It's so sensitive that if I just little bit, if I touch the board, everything gets rubbed off. Okay, so I'm having what? I'm having two molecules of CH3CHO, acetaldehyde. It is going to undergo reduction. The reducing agent will be NADH2. NADH2 itself will get oxidized. Right? Itself it will get oxidized but will reduce acetaldehyde into alcohol. Which alcohol? 2-carbon ethanol. See, this bond will break. One hydrogen over here, another hydrogen over here. So what will be produced? Two molecules of CH3CH2OH. And now this NAD which is produced is now again ready to be utilized during glycolysis. So the cycle also continues of NADH2 if fermentation is taking place. Correct? So NAD plus has been produced. It will go back to glycolysis. Again it will be reduced over there. If it is reduced, NADH2 will be produced. And if fermentation is continued, here again it will get oxidized. So same thing is going to take place. Right? So this is one type of fermentation called as alcoholic fermentation, where you reduce pyruvic acid into alcohol and your CO2 in two steps. First step is decarboxylation and second step is reduction. Okay, usually it is seen in case of yeast. Next, lactic acid fermentation. Lactic acid fermentation is also a type of fermentation, but it is a one-step process. First of all, here we are going to reduce pyruvic acid into lactic acid. Right? It is done with the help of bacteria, homofermentative bacteria like lactobacillus. Okay, this type of fermentation even take place in your muscle cells when there is insufficient supply of oxygen in muscle cells, when you are doing extreme level of exercise. Then what happens? Lactic acid fermentation take place. Now see what happens during this process. Now you are having two molecules of pyruvic acid. Simply reduction will take place. You know reducing acid is NADH2. Reducing agent is NADH2, which was produced during glycolysis. It will get oxidized into its original state, producing protons and free electron. In the presence of the enzyme lactate dehydrogenase, what will happen? You can see this double bond, it will break. One hydrogen, one hydrogen. क्या बन जाएगा? CH3CHOH COOH, and this is known as lactic acid. So this is how lactic acid is obtained in one step in your muscle cells or when lactobacillus bacteria is fermenting, right? And again, this NAD is ready to be utilized during glycolysis. So this is all about fermentation. I hope you have understood anaerobic respiration, where only two steps are taking place: glycolysis and fermentation, be it alcoholic, be it lactic acid. Now, moving ahead, anaerobic respiration is done. Now you have to see fate of pyruvic acid if oxygen supply is there. And this is very important. This takes place usually in your cell to generate energy. So next heading is aerobic respiration. The initial pathway is common, glycolysis. In the cytoplasm of the cell, in the absence of oxygen, 6-carbon compound glucose will break down into 2 molecules of pyruvic acid. Now if oxygen is there, so suppose this is a cell, right? This is the mitochondria inside the cell, and this is the cytoplasm. This is the matrix of mitochondria. This is a cell. Now in the cytoplasm, what took place? Glycolysis. So pyruvic acid, 2 molecules. 2 molecules of ATP, 2 molecules of NADH2, and 2 molecules of water are produced as end product of glycolysis, right? Now water will be released out as a byproduct. ATP cell will use. NADH2 we are leaving it for time being. Let's keep NADH2 still in the cytoplasm. We will use it later on. But right now our main focus is to understand what happens with these 2 molecules of pyruvic acid. Okay. Now these 2 molecules of pyruvic acid, if oxygen is there in the cell, then these 2 molecules of pyruvic acid from the cytoplasm will move inside the matrix of mitochondria. Correct? So these 2 molecules of pyruvic acid in the presence of oxygen will move inside the matrix of mitochondria, and further breakdown, further oxidation will take place in the matrix itself. So fate of pyruvic acid in case of aerobic respiration is taking place within the matrix. Now your pyruvic acid, if it has come into the matrix, the first step of breakdown of oxidation that takes place in the matrix is known as link reaction, also known as gateway reaction, right? Also known as gateway reaction. Now see what happens during this gateway or link reaction and why it is called so. Chemically, link reaction or gateway reaction is oxidative decarboxylation. So 2 molecules of pyruvic acid which has entered inside the matrix will first undergo oxidative decarboxylation in the presence of the enzyme pyruvate dehydrogenase. Now see, pyruvic acid which is formed during glycolysis in the cytoplasm after it has entered in the mitochondrial matrix will first undergo oxidative decarboxylation with the help of the enzyme pyruvate dehydrogenase, right? Chalo, so you have read this line I have taken from NCERT. Now I am going to write this 2 lines in the form of equation. Okay, before telling you about the equation, let's discuss about this complex enzyme pyruvate dehydrogenase. Students, pyruvate dehydrogenase is basically a complex enzyme which is made up of 3 components, E1, E2 and E3. It's actually a group of enzyme. So it's called as a complex enzyme, right? And each complex or each subcomponent of this enzyme has some or the other role. So we say that pyruvate dehydrogenase is a complex enzyme made up of 3 subcomponents, and each subcomponent has some or the other role to play during link reaction. Thick? Have you understood this much? Chalo. Now you are having what? You are having 2 molecules of pyruvic acid in your hand. In the matrix of mitochondria. What will happen? 3 things are going to happen with this pyruvic acid. Number 1, decarboxylation, removal of CO2. Number 2, oxidation. Again, the oxidizing agent will be NAD positive. NAD positive will extract protons and electrons from this pyruvic acid and will oxidize into its respective compound. Third, addition of coenzyme A. So 3 things are happening with the help of the same enzyme pyruvate dehydrogenase. ठीक, तो क्या क्या 3 चीजें होंगी? Number 1, there will be removal of CO2. Decarboxylation, it is done with the help of E1 component of the enzyme. Number 2, oxidation. The oxidizing agent will be NAD positive. It will get reduced into NADH2. This is done with the help of second component of the enzyme. And third function is addition of coenzyme A, and this is done by the third component of the enzyme, right? Chalo, so let's write this all in equation form. Coming to this page, okay, there's a Krebs cycle page. So where I should write, I'll just remove this and then I'll write the equation. So I hope you have seen that what are the 3 steps that has to take place with this pyruvic acid. So we are having 2 molecules of pyruvic acid, to which we are going to oxidize them with the help of oxidizing agent NAD positive. 2 molecules will be required. Along with this, coenzyme A will be required on the reactant side, right? Now enzyme is pyruvate dehydrogenase. Clear? Now in the presence of this enzyme, 3 activities are going to take place. First, there will be removal of CO2. 2 molecules of CO2 will be removed. Second, NAD positive will get reduced to NADH2 plus whatever product will be produced to that coenzyme will be added. So finally you obtain 2 molecules of acetyl coenzyme A. So 2 molecules of pyruvic acid gets oxidative decarboxylized to produce 2 molecules of acetyl coenzyme A. This is the product that is obtained during link reaction, and now this product is going to become the primary substrate for Kreb cycle. Now this reaction is known as link reaction because it is attaching glycolysis to Kreb. It is actually converting the end product of glycolysis to the first product of Kreb cycle. You are converting pyruvic acid into acetyl coenzyme A. Why? Because in Kreb cycle, pyruvic acid cannot be utilized. Acetyl coenzyme A will be utilized. And for that, you need to convert first PA into acetyl. Hence, you say that this link reaction or gateway reaction is joining glycolysis to Kreb cycle. It is just a intermediate reaction between 2 major processes of aerobic respiration. I hope all the 3 steps that how we convert pyruvic acid into acetyl coenzyme A is clear. Now we are going to start the next process in aerobic respiration, that is Krebs cycle. The substrate which is going to enter in Krebs cycle or which is going to participate in Krebs cycle is a 2-carbon compound acetyl coenzyme A. This acetyl coenzyme A, from where you obtained, dear students? You obtained after link reaction. So link reaction, as I told you, also known as gateway reaction, because it joins the two major processes of aerobic respiration, that is glycolysis and Kreb cycle. Glycolysis produces 2 molecules of pyruvic acid. From these 2 molecules of pyruvic acid, after the chemical reaction oxidative decarboxylation, 2 molecules of acetyl coenzyme A are obtained in the matrix of mitochondria by the process of link reaction. Now these 2 molecules of acetyl coenzyme A, for complete oxidation, will participate in Kreb cycle. This Kreb cycle also takes place in the matrix of mitochondria itself. So nowhere acetyl coenzyme A has to move. It will remain in the matrix and will undergo further breakdown, further oxidation. And the sequence, whatever it is going to happen with acetyl coenzyme A, that's all those sequence of steps together is known as Kreb cycle. Why do we call this as Kreb cycle? Because it was discovered by Sir Hanslet Kreb. So just to honor the scientist who has discovered the entire pathway for the oxidation of acetyl coenzyme A, we call this cycle as Kreb cycle. Correct? So what is Kreb cycle? You got it, that with the help of this Kreb cycle, we are going to oxidize acetyl coenzyme A to release CO2 and water. Right? Now, who discovered this cycle and where it is taking place? So it was discovered by Sir Hanslet Kreb, and it takes place in the matrix of mitochondria. What is going to be the first substrate of Kreb cycle? Then your answer will be a 2-carbon compound acetyl coenzyme A. Right? Chalo. So let's start. First, in the matrix of mitochondria, pyruvic acid entered, right? Which has undergone oxidative decarboxylation. That means removal of CO2, addition of coenzyme A, and reduction of NAD positive into NADH2. Right? What did you obtain after this? From the 3-carbon compound, then we obtained the first substrate of Kreb cycle, which is a 2-carbon compound acetyl coenzyme A. Correct? Now, this is the primary substrate, that is acetyl coenzyme A, which joins the primary acceptor of this substrate is a 4-carbon compound, that is oxaloacetic acid. So a 4-carbon compound oxaloacetic acid joins with the 2-carbon compound acetyl coenzyme A to obtain the first stable product of Kreb cycle, a 6-carbon compound called citric acid. Now, since the first stable product is a 6-carbon compound, therefore this cycle is also known as citric acid cycle. Kreb cycle can also be known as citric acid cycle. Clear? It is a 6-carbon compound. This is simply addition reaction taking place in the presence of the enzyme citrate synthase. Right? So in the presence of the enzyme citrate synthase, 4-carbon compound oxaloacetic acid joins with 2-carbon compound to produce citric acid. Now, this citric acid will undergo isomerization. Isomerization means, now this citric acid will convert itself into its isomeric form. Now, this conversion of citric acid into isomeric form, my dear students, it's a 2-step process. First, citric acid will be converted into cis-aconitic acid, right? Cis-aconitic acid, and then this cis-aconitic acid will finally get converted into the isomeric form of citric acid, which is isocitric. Right? So these two steps of the reaction, they are catalyzed by the enzyme isomerase. Why isomerase? Because isomerization reaction is taking place, and that is done in two steps. First, citric acid convert itself into cis-aconitic, and then this cis-aconic get converted into isocitric acid. Now, the first three intermediates which you can see, all these three are 6-carbon compound. Correct? Moving ahead. Now, this isocitric acid will not stop, because our aim is to completely oxidize the 2-carbon compound acetyl coenzyme A into 2 molecules of CO2. So first step after isocitric acid formation will be oxidation of isocitric, and the oxidizing agent which will oxidize isocitric is again NAD positive. No oxygen addition, simply removal of hydrogen and electron will take place from isocitric acid, that will be accepted by the oxidizing agent NAD positive to form NADH2. So NAD plus will get reduced. In return, it will oxidize your isocitric acid to produce another 6-carbon compound, the oxidized form of isocitric, that is oxalosuccinic acid. Correct? This is also a 6-carbon compound. Now, so far you have obtained 4 intermediates, and all these 4 are 6-carbon containing compound, and in each compound there are 3 carboxyl groups. If you see the structure of all these 4 intermediates, then you will find that in each structure there are 3 carboxyl group, tricarboxylic acids. So all these 4 intermediates are actually tricarboxylic acid, and hence Kreb cycle can also be known as TCA cycle. It is known as citric acid cycle because the first stable product is citric acid. This cycle is also known as TCA cycle because the first 4 intermediates are tricarboxylic acids. Clear? Now, this 6-carbon compound, oxalosuccinic acid, my dear students, will now undergo decarboxylation. Will now remove the first CO2 of acetyl coenzyme A, first carbon in the form of CO2, to produce another intermediate, that is alpha ketoglutaric acid. This is a 5-carbon compound which is produced after decarboxylation of oxalosuccinic acid, or you can say simply oxidative decarboxylation of isocitric. And these two reactions, my dear students, they are catalyzed by the enzyme isocitrate dehydrogenase. Isocitrate dehydrogenase enzyme catalyzes the conversion of 6-carbon compound isocitric into 5-carbon compound alpha ketoglutaric in 2 steps. Now you are having 5-carbon alpha keto. This will again undergo oxidative decarboxylation, that means removal of CO2, production of NADH2, plus there will be addition of coenzyme A. See, here one coenzyme A was removed. Now the same coenzyme A which was removed on the first step will come and get attached to this compound. So first addition of coenzyme A, removal of CO2, plus reduction of NAD positive, or you can say oxidation of alpha ketoglutaric acid. So here I am writing as I am not having space. To produce what? After these 3 steps, the next 4 carbon compound that will be produced is succinyl coenzyme A. So this is now a 4 carbon compound produced by the oxidative decarboxylation of alpha ketoglutarate acid. Right? Now enzyme which is required is alpha ketoglutarate dehydrogenase. Fine. Now this succinyl coenzyme A, my dear students, is a high energy intermediate. It is having number of phosphate groups attached to it. So one of the phosphate group, terminal phosphate group from succinyl coenzyme A will now be removed. So what will be the next step? Removal of one of the terminal phosphate of succinyl coenzyme A, that will be accepted by GDP. GDP will accept the phosphate and will produce another energy formula called GTP. Like ATP, GTP is also energy source, CTP, TTP, all these are energy sources. So this again becomes substrate level phosphorylation, because with the help of substrate you are obtaining one of the form of energy, and itself it will get converted into succinic acid, which is again a 4 carbon compound. See, now my dear students, focus. You had 6 carbon in citric acid, and the 6 carbon were actually of 2 carbon from acetyl and 4 carbon from oxaloacetic. Now oxaloacetic is only helping acetyl coenzyme A to get completely oxidized into CO2. That means in whole Krebs cycle, if 2 CO2 are coming out, those 2 CO2s are representing the complete oxidation of acetyl coenzyme A. That we have done. See, first CO2 over here and second CO2 over here. Now we are left with 4 carbon compound. Now this 4 carbon compound, to maintain the continuity of the cycle, so that if second acetyl coenzyme A comes, it also gets the oxaloacetic acid for further reaction. Regeneration of this compound is must. Right? Now whatever we are going to do, conversion of succinic acid now will take place to regenerate OAA, so that continuity of the Krebs cycle can be maintained. 2 carbon of acetyl coenzyme A, I am repeating, please listen. 2 carbon of acetyl coenzyme A you have already oxidized to produce CO2. Now 4 carbon which are left are actually of OAA. We cannot remove them in the form of CO2. Why? Because we want to regenerate this compound to maintain the continuity of the cycle. So whatever now reaction will take place, that will take place towards the formation of OAA, so that the cycle can be completed. Now in that case, this succinic acid will now be converted into another 4 carbon compound called fumaric acid. Right? Now conversion of succinic acid into fumaric acid is also an oxidation reaction, but here the oxidizing agent is another redox equivalent FAD positive, that itself gets reduced to FADH2, in return oxidizes succinic acid into fumaric acid, and this reaction takes place in the presence of the enzyme succinate dehydrogenase. Clear? Fumaric acid is then converted into malic acid, and in the final step malic acid is converted into oxaloacetic acid by undergoing oxidation. And here the oxidizing agent is going to be NAD positive, that itself gets reduced to produce NADH2 but oxidizes malic acid to regenerate oxaloacetic acid. So these are the intermediates. So many intermediates are produced just for the oxidation of acetyl coenzyme A and regeneration of OAA. Correct? Yes or no? So this is how the cycle runs. Now the cycle is actually going to run two times for one glucose molecule. Why? Can anybody answer me that why it is going to run two times for one glucose molecule? The simple answer is that since one glucose molecule produces 2 pyruvic acid, from 2 pyruvic acid to acetyl coenzyme A, and for 2 molecules of acetyl coenzyme A, the cycle is going to run 2 times. Right? So this is how the cycle runs. I hope you have understood the nature of Krebs cycle and how many intermediates are produced and whichsoever enzymes are used. Okay, now we are going to check the result of Krebs cycle as you check the result of what? Of glycolysis. So if I talk about the result of Krebs cycle, then what are the things you obtained? Dekho, per Krebs cycle if I say, then we actually obtained 2 molecules of carbon dioxide. Yes or no? From 2 acetyl coenzyme A, 2 times decarboxylation took place. So 2 CO2 you have obtained. Apart from that, we have obtained 3 NADH2. Check this. Where are those 3 NADH2s? Let's mark them. Number 1, NADH2, oxidation step. Second step and third step. 3 NADH2 we have obtained, 1 FADH2 we have obtained, and 1 energy source. This reaction is known as SLP. Don't forget that. And 1 GTP we have obtained per Krebs cycle. So 2 CO2, 3 molecules of NADH2, 1 molecule of FADH2, and 1 direct source of energy in the form of GTP. These things we have obtained per Krebs cycle. But actually for 1 molecule of glucose, this cycle is going to run 2 times. So everything will be done into 2. Hence 4 CO2, 6 NADH2, 2 FADH2 and 2 direct source of energy in the form of GTP. These are the end products of Krebs cycle. Now my dear students, if you see that your 6 carbon glucose has now been actually converted into 6 CO2. How you have converted? 4 CO2 has been removed in Krebs cycle and 2 CO2 were removed during link reaction. In link reaction also oxidative decarboxylation was taking place. So actually overall we have completely oxidized glucose into CO2. It has been completely broken down into its simplest form. Now you must be thinking, ma'am, complete oxidation has been done, but there are 2 problems. One, what about your ATP? In the beginning of the chapter you said, ma'am, that about 36 or 38 ATP will be obtained, but so far we have only obtained 4 ATP, 2 over here and 2 in glycolysis, if you remember everyone. So ma'am, only 4 ATP we have obtained and you are saying complete oxidation is done, plus no oxygen has been used. Wherever you are doing oxidation, you are just removing proton and electron. You are nowhere oxidizing with the help of oxygen. So why it is called as aerobic respiration? It should be also called as anaerobic respiration. So I hope every student must be having these 2 trouble, and that where are the remaining ATP and where is the use of oxygen if you are saying that complete oxidation has been done. So all my dear lovely students, not to worry, both the questions will be answered in the next segment of this chapter, Electron Transport System. In electron transport system, you will see the use of oxygen as well as the remaining generation of ATP. So my dear students, aerobic respiration is yet not over. Maybe glucose oxidation is done. Maybe 6 carbon of glucose has been converted into its simplest form. But still the process is not over, because your 2 questions are still left unanswered. So one by one, I'll show you the answers of both your questions. Now, you are thinking about ATP. So if you know that during the whole process, apart from 4 ATPs, 2 in glycolysis and 2 in your Kreb cycle, apart from these 4 ATPs, you have also produced 8 molecules of NADH2. Rather, I will say, 10 molecules of NADH2. How? 6 from Kreb cycle, 2 from link reaction and 2 from glycolysis. So total 10 NADH2 you have and 2 FADH2 also you have. So these compounds 10 NADH2 and 2 FADH2 are actually your indirect source of energy. These reduced form of NAD and FAD, when they will return back to their original state, when we are going to oxidize them, then during their oxidation a proton gradient will be developed across the inner membrane of mitochondria. That will be utilized by the enzyme ATP synthase to generate the remaining ATP. And that step to generate remaining ATP by the oxidation of NADH2 and FADH2 is known as electron transport system. So shall we start electron transport system? Yes, ma'am. Okay, very good. So our next topic is ETS. The same manner you studied in chloroplast, chemiosmotic theory. So here also we are going to study chemiosmotic theory only, which is responsible for ATP generation. You know there are 4 requirements. Number 1 requirement is a living membrane. So here the living membrane is going to be cristae, that is the inner membrane of mitochondria. Second important thing that we require was proton pumps. So on the inner membrane of mitochondria, different proton pumps in the form of complexes, complex 1, 2, 3, 4 are present, that will pump the proton from the matrix in the intermembranous space, because of which the concentration of proton in the IMS will increase and in the matrix will decrease, creating a gradient, right? Apart from this, on the membrane, one more complex is present called complex 5, that is also known as ATP synthase. And this ATP synthase enzyme is going to break the gradient by passing the proton from the IMS back into the matrix, and while doing this, ATP will be generated. Clear? So this is whole what we are going to do, and with this your aerobic respiration will also be done. Oxidation of glucose, tick mark. Now let's see generation of remaining ATPs. Chalo, focus. So first point you have all understood. Second point, what is the aim of ETS? Its aim is to generate energy by the oxidation of already reduced NADH2s and FADH2 with the help of complexes found on inner membrane, right? Now, principle क्या है ATP synthesis का? That is breakdown of proton gradient and then generate energy. Okay. Fourth, now this method of ATP formation, which method? That is generation of ATP by the help of proton gradient by performing oxidation of some reduced equivalents is known as oxidative phosphorylation. So there are 3 methods of producing ATP. One is substrate level phosphorylation, second is oxidative phosphorylation, and third is photophosphorylation, which you study in chloroplast, right? Okay, moving ahead. Now what are those complexes on the inner membrane? So suppose this is my inner membrane of mitochondria, right? Now, towards the inner side of the inner membrane is present matrix, and towards the outer side is present intermembranous space. You all know the structure of mitochondria. Cell chapter has been already done. Okay, so on this inner membrane are present the various complexes. First is complex 1, that is known as NADH2 dehydrogenase. Correct? This complex is made up of two components. What are those? One is, so the first component is iron sulfur, and the second component of complex 1 is FMN. Talking about complex 2 is made up of again two components. The two components for complex 2 are iron sulfur as well as FAD. Complex 2 is also known as succinate dehydrogenase or FADH2 dehydrogenase. This complex 2 is actually the enzyme which is also utilized in Kreb cycle. Third, complex 3, as 3 says, so it is made up of three components: cytochrome B, cytochrome C1 and iron sulfur. So iron sulfur, cytochrome B, cytochrome C. Right? Fourth complex is cytochrome C oxidase, which is again made up of four components. Complex 4, four components. Simple to remember. The four components of complex 4, cytochrome oxidase, C oxidase, is copper A plus, copper B plus, cytochrome A1 and cytochrome A3, right? And the fifth component is nothing but your complex 5, that is ATP synthase enzyme made up of two components, F1 and F1 ATPase. F0 and F1 ATPase. F0 is actually the channel through which the protons are going to go back, and while going back they will face the bulged part of the enzyme called F1 ATPase, which is bulging towards the matrix of the mitochondria. So this is F1 ATPase, and this together, this complex part is known as complex 5, the enzyme ATP synthase. I hope you have understood whatever is written on this page. So let's, without delay, let's start with the process of ATP synthesis by oxidative phosphorylation method on the inner membrane of mitochondria. So here I am going to draw the inner membrane of mitochondria, but before that you should know that there are two equivalents to which you have to oxidize: NADH2 and FADH2. For the oxidation of NADH2, complex 1, 3 and 4 are involved. 1, 3 and 4 are involved for the oxidation of NADH2. If I talk about the oxidation of FADH2, students, then complex 2, 3 and 4 are involved. That means 3 and 4 are common, but complex 1 is specifically for the oxidation of NADH2 and complex 2 is specifically for the oxidation of FADH2. So here I am going to show you the oxidation of complex, here I am going to show the oxidation of NADH2 by using complex 1, complex 3 and complex 4. चलो, तो सबसे पहले हम ड्रॉ करते हैं inner membrane को. So this hole is the inner membrane of mitochondria on which your proton pumps in the form of complexes are present. This is the region present within the inner membrane called matrix, and this is known as intermembranous space, that is the region outside the inner membrane. Correct? Now see what will happen. On this inner membrane, complexes are present. How many? All the complex. But here I am only going to show you 3 complexes that are required for the oxidation of NADH2. So I am going to miss out complex 2. So this is my complex 1, which is made up of 2 components, FMN and FES. Remove complex 2 for time being as it is not required. Then comes your complex 3, which is made up of 3 components, that is iron sulfur, cytochrome B1 and cytochrome C, followed by complex 4 made up of 4 components, that is cytochrome A1, copper A plus, cytochrome A3 and copper B plus. Correct? Followed by complex 5 made up of 2 components, F0 channel which is embedded in the membrane and the matrix, and the F1 ATPase that is facing towards the matrix. So this is your F0 channel and this is your F1 ATPase, together called as complex 5. Now, living membrane you have seen, proton pumps you have seen. Now to generate energy, gradient is required. So now let's see how gradient is developed. For gradient, first oxidation of the reduced complexes has to be done. So the reduced equivalents are NADH2 and FADH2. First I am going to oxidize NADH2 with the help of complex 1. Complex 1 oxidizes all the 10 NADH2s one by one present in the matrix, right? 8 NADH2s were already there in the matrix. 2 NADH2 which were produced during glycolysis in the cytoplasm, they also move in the matrix. In all, now there are 10 NADH2 in the matrix which need to get oxidized with the help of complex 1. One by one they get oxidized. So first of all, if first NADH2 comes and it gets oxidized into its original state, so what will it produce? NAD positive plus 2 protons and plus 2 electrons. Correct? My dear students, this complex 1, what it does, see, trouble is with electron. What happened with the electron? Once oxidation of NADH2 is done into its original state, that is NAD positive, it releases protons and electron. Electron, you know, cannot be released in the membrane directly. If electrons are produced, then there should be, if they are produced from one component, then it should be sent to the second component. You cannot release electron freely in the membrane. Correct? So these 2 electrons which are produced by the oxidation of NADH2, they are soon accepted by the first component of FMN, that is of complex 1, that is FMN, flavomononucleotide. So the electrons released by the oxidation of NADH2 are accepted by the first component of complex 1, that is FMN. FMN, when accepts the electron, it is not happy. It says, I want to release these electrons, so it soon passes these electrons to the next electron acceptor within the complex 1, that is iron sulfur. Iron sulfur, for time being, it takes the electron from FMN, it gets reduced, but it is not happy. It says, I also want to pass these electron, I cannot keep it for long. So to rescue FES in complex 1 comes another electron and proton carrier, ubiquinone. Ubiquinone, my dear students, these are freely moving electron and proton carriers on the inner membrane. These ubiquinone are simply free, mobile. They can move anywhere here and there on the membrane. They are both electron as well as proton carriers. So they accept the electron from iron sulfur as well as the protons. You see, NADH2 also produce 2 protons. So these accept. So ubiquinone also accept the proton from NADH2. Now, after accepting electron and proton, it gets reduced to ubiquinol, UbQH2. Correct? But will it like to stay in this phase? Is it happy to stay like this? No. It says I want to convert myself into my original state. So what it shall do? It shall pass those electron and proton to the next complex, that is complex 3. But complex 3, the components of complex 3 are only electron acceptor. They will accept the electron from ubiquinone, but they will inhibit to accept proton. So what this ubiquinone does, it passes those protons in the IMS. It acts like a proton channel and passes the proton of the matrix into the IMS and remaining electron to the first component of complex 3, that is iron sulfur. Right? Meanwhile, my dear student, complex 1, while it is oxidizing NADH2, while it is passing electron, it also pumps 4 proton. It is also a proton channel. It is also a proton channel. So it pumps 4 proton from the matrix in the IMS. Meanwhile, it is oxidizing NADH2, passing electron. So meanwhile, this function, it is a proton channel. So the channel is open, it is functioning. So 4 protons slips from the matrix in the IMS. 2 proton already there, 4 came more. So can you see the gradient has been developed, is started developing. The matrix proton, they are decreasing, and in the IMS the number of protons are increasing. Can you see gradient has been started? Okay, now where are electron? Electron are with complex 3. Iron sulfur of complex 3 says, no Baba, I am not happy with this electron, please take away. So it passes the electron to another component of complex 3, and then again it passes the electron to another component of complex 3. Finally, when the electron comes to the last component of complex 3, this component says, I am not happy, I am not going to keep this electron for long, please take this electron away from me. So now what will happen? Now this electron from cytochrome C of complex 3 has to be passed to complex 4, but they cannot do it directly. There is one more mobile electron carrier which is attached towards the outer side of the inner membrane called cytochrome C, a small protein moiety which is attached on the outer side of the inner membrane that is facing the IMS. There is a small protein moiety called cytochrome C that accepts the electron from this cytochrome C of complex 3 and passes those electrons to complex 4. So this is like a intermediate. This protein moiety which is attached towards the outer side of the inner membrane, this acts like a connector between complex 3 and complex 4. So now the electrons are with complex 4. The component of complex 4, cytochrome A1, accepts the electron, but as the tradition says, it is not going to keep it. So it passes electron, passes electron, passes electron, and finally the electron from NADH2 has come to the last component of complex 4. Now from here, those two electrons will be accepted by terminal electron acceptor oxygen, which along with the 2 protons in the matrix will produce water, that is one of the byproduct of respiration. Clear? Can you all see that what is the role of oxygen? Where is the role of oxygen? So far in complete aerobic respiration, you have studied glycolysis, no role of oxygen. Link reaction, no role of oxygen. Kreb cycle, no role of oxygen. Complete oxidation done in the absence of oxygen. But at the last step of ETS, oxygen is required. It is the terminal electron acceptor. You can also say that this oxygen is going to drive the entire process. If here oxygen is not available, gradient will go in vain, electron will be released as it is in the membrane, in the matrix, and your whole process will go in waste. No ATP generation, no extra ATP generation will be done. So oxygen is very, very important. That is why this type of respiration is known as aerobic respiration. Correct? So water has been obtained, right? Similarly, like complex 1, complex 4 is also a proton channel, and while passing electrons, it also passes 2 protons from the matrix in the IMS. Can you see the complete electron transportation has been done? NADH2 has been oxidized, right? Its 2 electrons has been received by oxygen to produce water, and all the proton has also been passed into the IMS, creating a proton gradient. Correct? How much proton gradient has been developed? Of 8 proton. 1 NADH2, when it gets oxidized, 8 protons, they come in the IMS. Now these 8 proton wants to go back. They wants to go back from where they came, that is to the matrix. Correct? Yes or no? So 8 proton in total wants to go back to matrix, so that ATP, so that this gradient can be finished off and ATP generation can be done. So every time one pair of proton, that means 2 proton, they go back in the matrix generating 1 ATP, right? So how many pair of protons are there? 4 pair. So how many ATP can we generate? 4 ATP. So can we say that one molecule of NADH2 can produce 4 ATP? Is this correct? No, not correct. One NADH2 is responsible for producing only 3 ATP. How, ma'am? Right now you are saying 8 protons are there in the IMS. That means 4 pair. All these pair will go back, and when one pair will go, 1 ATP. So logically, mathematically, 4 pair, 4 ATP. So where is this 1 ATP ka locha? Where is this 1 ATP going? So students, pay attention. 3 pair of proton, they pass through the enzyme producing 3 ATP. But 1 pair of proton, it does the cheating and it passes the membrane directly without the help of enzyme. It uses certain chemicals present on the membrane called uncouplers. So it uses those chemicals on the membrane and they pass as it is to the matrix, breaking the gradient but not generating energy. So simply you have to remember that out of 8 proton, 6 proton, that means 3 pair, they passes. Out of 8 proton, 6 proton, they pass through the enzyme producing 3 ATP, and 1 pair of proton gets in vain. It does not produce energy. Clear? Yes or no? So how many NADH2s do we have? We have 10 NADH2. And of all these 10 NADH2s will get oxidized, then how many ATPs we will produce? We will produce 30 ATP. So 4 ATPs you were already having. Yes or no? This is how ETS runs for NADH2 oxidation. Now if I talk about FADH2 oxidation, then whole process remains the same. It's just complex 1 get replaced by complex 2. And one thing you have to remember, complex 2 does not pumps any proton. So there these 4 proton gets missing. Hence, during the oxidation of one molecule of FADH2, a proton gradient of 4 proton is created, not 8. Unlike that of NADH2, when one molecule of FADH2 gets oxidized, then proton gradient of 4 proton is only generated. That means 2 pair, and both the pair, they go through the enzyme producing 2 ATP. So we can say that one molecule of FADH2 yields 2 ATP and one molecule of NADH2 yields 3 ATP. Right? Now, how many FADH2s and NADH2s are there? There are 2 FADH2s and 10 NADH2s. So, total 4 ATP from FADH2 and 30 ATP from NADH2. Total 34 ATP you are obtaining by the process of oxidative phosphorylation, which you can also called as indirect ATP, right? And 4 ATP, you know, already you have 2 in the form of GTP through Kreb cycle plus 2 ATP in the form through glycolysis, right? So that makes plus 4 ATP, and total energy balance comes to be 36 ATP. That means, sorry, 38. 38. That means when one molecule of glucose is getting oxidized aerobically, then it can produce 38 ATP. 34 indirect and 4 direct. Hope you have understood this energy balance sheet. Hope you have understood what is aerobic and anaerobic respiration. This is the major part of this chapter. If you have understood this, then we are just moving towards the end of the chapter, where we have to study 2 major topics, amphibolic pathway and respiratory quotient. First, talking about amphibolic. In the starting of the class, I told you that respiration is neither catabolic nor anabolic. Rather, we say that it is amphibolic. Amphibolic means what? It is both catabolic and anabolic. How catabolic and how anabolic? Let's try to justify. Bachcho, it is catabolic because overall you see a complex substance like glucose is broken down into its simplest form, that is CO2 and water. Right? So complex substance like glucose is broken down into its simplest form. Hence this reaction is catabolic. This we know since our childhood. But why are we calling it as anabolic also? Because while this oxidation, while this conversion of complex into simpler substances is taking place, there are so many intermediates produced, like succinyl coenzyme A, which you have seen in Krebs cycle, then alpha ketoglutaric acid, pyruvic acid. So many such intermediates are produced during this oxidation, which can be used further for synthesis of some important compounds like alpha ketoglutaric acid. In mineral nutrition chapter, you have already seen it is used for the formation of proteins like glutamic acid, right? So parallelly catabolism is taking place, and parallelly some anabolic reactions are also taking place. Hence this respiration is both catabolic and anabolic. Overall it is catabolic, but in between some anabolic reactions can also take place, like for example one of the intermediate alpha keto producing proteins like glutamate. Correct? So that is why respiration is both catabolic and anabolic, called as amphibolic. I hope I am clear. More examples are there that you can learn it from your NCERT. Now moving ahead with the next and the last topic, that is respiratory quotient. Respiratory quotient means what? Respiratory quotient simply means the ratio of the volume of CO2 evolved to that of oxygen consumed during respiration. You can simply write RQ as volume of CO2 evolved during respiration upon the volume of oxygen consumed. Correct? So volume of CO2 evolved upon volume of oxygen consumed is RQ. Very simple example I will use to explain this. For example, if I talk about carbohydrates, for all carbohydrate, RQ value is always 1. Why? What is the reason? Let's see. Now, the simplest form of sugar is C6H12O6. Oxygen is required to completely break down into its simplest form. Now just balance this equation. Balanced. How many volume of CO2 is required? 6. How many volume of oxygen is required? 6. So 6 by 6, 1. Hence RQ of any sugar you take, it is always going to come 1 when you balance the equation. Similarly, for fats, RQ is less than 1, that is about 0.7. For proteins, it is about 0.9. Correct? So respiratory quotient depends upon the type of respiratory substrate. Like for example, for carbs, RQ is 1. If I talk about for proteins, it is 0.9. For fats, if I talk about, it is 0.7. One example has been quoted in your NCERT about tripalmitic acid. It has been proved that in that case, the RQ value is 0.7. For any fat you talk about, every time RQ value is constant, that is 0.7. Sometimes your respiratory substrates can also be organic acid. Like for example, malic acid and oxalic acid. For malic acid, the RQ is about 1.33. One more you shall learn. Oxalic acid, it is 4. So please, you can learn these two RQs of organic acid, which are usually asked in NEET. If you learn this much, more than sufficient. No question will be asked apart from this. So with this, finally we have come finally come to the end of the section, and before entering into thank you slide, let's do some questions. The first question says, to maintain the continuity of Krebs cycle, regeneration of which compound is required? Your options are oxalosuccinic, oxaloacetic, acetyl coenzyme A, or more than one option. Just now I told you that regeneration of OAA is must to maintain the continuity of Krebs cycle, as the primary acceptor in Krebs cycle is OAA. So the answer of this question becomes B. Yes or no? Moving ahead. Next question. What is link reaction? Now you have been given some 4 statements. You have to choose which one is right and not. First, also known as gateway reaction. Correct. Does it occur in the cytoplasm? No. Rather it occurs in the matrix of mitochondria. So B statement becomes wrong. Next statement. Chemically it is oxidative decarboxylation. Yes, pyruvic acid undergoes oxidative decarboxylation to produce acetyl coenzyme A in the presence of the enzyme pyruvate dehydrogenase. So this statement is also right. Hence the answer of this question will become D. That is, more than one statement is true. Next question. Glycolysis is also known as EMP, citric, tricarboxylic, all of the above. So, bachcho, these two are the name of Krebs cycle. Why? Because in Krebs cycle, the first stable product is citric acid, and during Krebs cycle, there are 4 intermediates which are tricarboxylic in nature. However, glycolysis is also known as EMP pathway after the name of the scientist who discovered it, Embedon Meyerhoff Parnas, EMP. Hope you have understood everything. And with this, we come to the end of the chapter, respiration. Very, very important, easy, quick to understand, quick to learn, only if you pay attention. So, my dear students, it's a humble request rather to say that please study well, stay focused, keep working hard. See you in the next video. Till then, bye-bye everyone. Take care.